this paper just dropped, and it's on stem cells. And we're going to get into what the heck stem cells are and why these are important. But the essential crux is the group that published this paper has already received the Nobel Prize. They received the Nobel Prize in 2012 for their ability to demonstrate that you can reprogram differentiated, so like finalized cells, like your skin cells. You can reprogram them to be stem cells by just turning on four different genes. You can make them be stem cells. In this paper, what the researchers did was they took out two of the genes inside mouse stem cell or mouse cells in mouse cell culture, not stem cells, but regular differentiated cells. They took out two of those genes and they replaced them with two genes from a single-celled organism called the cyanoflagellate. And the cyanoflagellates also have these two genes. They have the genes SOX and POU, which we also have in our cells, but they replaced the mouse version with this single-celled animal version. This single-celled animal has one cell and it's not a stem cell. Yet these genes are there and they're activated, which means that these genes probably do something different in these more ancient animals, but they're the common ancestor between all living animals. They're the most related between all living animals. We're going to dig into that in a moment too. Not switching out any genes. All they did was take mouse cells and they turned on four genetic elements. Of those four genetic elements, two of them were this SOX and POU. So it was not ethical determination in the sense of like, well, what's closest and what can we switch? It was they already had four genes. And then they saw that this really ancient common ancestor had two of them, and those two genes looked a little bit different. And so then they switched out those two same genes with the existing ones. They just swapped them out. And so everything still functioned just fine, but it reverted those cells to the stem cell-like state. Now, one thing that's important, I think, Jay, to take away from this is that they will need to eventually deactivate those genes. So you can imagine a situation where, let's say I take your skin cells and I grow a heart from your skin cells, which is like the end game, right? If you can go back and grow a full heart from your skin cells, then if you needed a heart transplant, you don't need to wait for an organ donor that you may or may not reject. You can just do same heart, but you need to be able to turn off those stem cells. You need to be able to, you make them back into this pluripotent stage where you can make anything, and then you need to have them turn back off. Because if they don't end up turning back off, you have uncontrolled cell growth, which is cancer. Hopefully that makes sense, Jay. And they were actually able to do that. The way they were able to do that and demonstrate this is that they went through, hold on, let me grab this quick figure summary. We'll look at the other figures in the paper too, but this is just kind of the straight shot image. Here we go. This is their workflow, Jay. It's a nice way of like demonstrating what exactly they did, and it's actually kind of easy to follow, which is great because usually these are not easy to follow. But they put in the genetic element that they wanted to switch out, and they even have a visual marker that they've put in what they wanted. So with lasers, they're able to demonstrate that these cells have in fact gotten whatever thing that they've wanted to pop in. So they wanted to have removed the existing gene and replaced it with this single cell animal's copy of the gene. And the way they are able to mark that is then the cells are glowing green. So if it's been a successful conversion, the cell goes from not glowing to glowing. And then you actually pick out the green cells, the glowing ones, and then you can culture them in a dish. And it turns out, so then the final step is this what's called pluripotency validation, which is if it's truly become a pluripotent stem cell, you should be able to grow whatever tissue you want to grow. Like, y'all, this could revolutionize regenerative medicine. That's how big of a finding this is. Because up until now, I mean, this group has won the Nobel Prize already for being able to make your regular cells stem cell-like. But this here, to make it pluripotent, we're just showing off this brand new paper that was highlighting the possibility of stem cells that are pluripotent, meaning that they can become any tissue. And the way this is done, it's you take regular cells from your skin and you replace two genes. Now, the researchers have found that 4 genes were necessary to turn on to make cells look kind of like a stem cell. And they actually had won a Nobel Prize for it in 2012. But now, their new study, they're still looking at those same 4 genes, but 2 of them, they pop out. They remove 2 of them, and they actually will replace them with a single-celled animal's genes copy. So these two genes exist in us, in mice, and this single-celled animal. They just look a little, the gene looks a little bit different between different animals, but it should be functionally the same thing. But when they replace this genetic element from that single cell animal and put it into the mouse cell, it reverts it back to a stem cell. And the real proof in the pudding here, they made a pluripotent pile of stem cells that they then implanted and it became a mouse. So like these mouse pups here, the way that you can tell which ones are the genetically edited ones, so you have to have a way of marking, right? Because you might not know which mouse is from who. These, normally the lab mice have these red eyes, but they put in a genetic change in their stem cells to make the animals that have been genetically modified to coming from that pile to have these black eyes. So the researchers took skin cells from a mouse and they replaced two genes. The two genes they replaced came from a single-celled animal they found in the ocean. This single-cell animal is the common ancestor to all animals today. We're going to look and see what it looks like in just a few minutes. Don't worry. They took its two of its genes. Two genes were soft. That pile of skin cells reverted to pluripotent stem cells. And those pluripotent stem cells were then implanted and they made a mouse. They grew a mouse, a brand new mouse. And the way that they marked what was genetically modified was the eye color. So normally these white mice for the labs have these red eyes for the albino marker. But here they have these black eyes. And the reason for that is they directly put in that genetic change so you can track what happens. So you could track, like, from this litter, this is the one that was from this pile of our pluripotent cells, and this is a regular embryo. So it's actually really cool because you have the comparison of the not genetically modified one and the regular and seeing if there's any difference. And yeah, so Jay, it's the exact same otherwise, right? It's the exact same. Like, it's 100% a mouse, and they could do some kind of microsurgery to transplant organs. Like, this would be an exact copy. I mean, mice are actually a bad example because you can buy genetic lines and lineages for labs where they're all already genetically identical. But let's say that they weren't. Then you could do this kind of procedure and actually help a mouse in need. It's really covered in the news yet because, and I don't know why, but I feel like this is like a big revolutionary change. Because again, imagine, I mean, it's not yet on the table for switching out organs, but if you need a new lung, one of the limiting factors so far is that we haven't had the cells to be able to grow new organs, right? We just haven't had these pluripotent stem cells that, I mean, you can get them when you're born, your umbilical cord has some. And so we actually banked Munchkin's umbilical cord blood and put it off into, and it's frozen away, right? So then those are her stem cells. So if they grew organs from those, they would be replaced, no problem. But not everyone has done that. Not everyone, like, Gers, you and I didn't have that technology when we were babies, putting away those, umbilical cord goo, as it were. So yeah, the idea would be that you can use this technology and help people. And I think that's amazing. That's the limiting factor for, as GK and Jay points out, we can't yet straight up grow an organ in a dish. And the reason for that, Jay, we don't have all the factors yet. So you can start having cells form a heart, but they don't really finish. So what you end up getting is a mound of heart cells, and it doesn't finish forming the full shapes. Mound of heart cells, but not the right shape. Got you. So there factors need to create the correct shape of the organ, Jay, and then once the shape is also made, how the blood vessels go throughout. So it's like they're ready, but they don't know their next step. And so that's still an issue here, right? But this at least allows us to make something that your body will not reject. So the issue is, right, if you get a heart transplant, I believe, correct me if I'm wrong, not all heart transplant actually will last your lifetime, where you might need another heart at some point. Yep, yeah, exactly. And they stop working at some point, Gers, but because they're immune suppressants, right? And eventually your immune system is like, all right, we figured this out. Away with a need for a transplant list, right? All you do is, well, all right, let me take Mr. Giraffe, let me take some of your skin cells. I will switch out these two genes, make your cells revert to stem cells, tell them to become whatever organ you need, and then, pop goes the weasel. We put it in you and you're Gucci. You're all on your way. That's like the dream scenario, right? There's still a ways to go there. But one of the big factors that was not necessarily stopping it, but really a hindrance is up until this point, it's like if we did have a population of stem cells, it was from a certain lab and it's not tailored to the patient. So there's still the chance of rejection. And this way, you can grow that organ to exactly match the person. And that would be really, really cool, right? That should be a much easier way to go. That you don't need to take the immune suppressants to prevent the rejection. And, you know, I mean, we're going down the line and like what I think could happen from it, but it is quite revolutionary. Do our bodies use stem cells naturally? So here's the huge difficulty in this. Stem cells have growth factors that are activated because what they do is they divide what are called asymmetrically. So, porkers. So, Jay, because they're dividing asymmetrically, meaning that, again, one cell, when it divides, stays a stem cell, the other one starts undergoing differentiation to become whatever it needs to become. That means there's constant growth. You have to be able to turn that off, because if you don't, you get a bunch of cell growth, which is cancer. So that is a double-edged sword. If you don't do it right, you will have more cell growth, and that'll cause a tumor. So you have to be able to turn it off. Now, the nice thing about this is if you hijack developmental programming, you're taking stem cells, you're putting it into the embryonic development cycle, and there you're going to activate genetic elements and turn everything off. Like right now, see, Jay, if we had just like loosey-goosey activated stem cells, like imagine if you sever your spine from a terrible accident and you can't use your lower half of your body. If you just, injected a little bit of stem cell into your lower back and you reform those nerve connections, right? Because you regrow those neurons, then you can walk again. But it's the turning off of those stem cells that our body, the mechanisms aren't activated, and they're not activated because our stem cells aren't activated. So if you turn on stem cells without the deactivator, then you're just going to have a tumor grow. And that's like the difficulty of this system as well, of like working with stem cells, because like their whole job is to grow, and you have to have regulators there that tell them when to grow, how much to grow, and when to stop. A sliver of a spine. Yep, exactly. Yeah. So it would be amazing to repair something. That's why, Jay, we're studying things like planarians, right? Which when you can, if you cut them in half, a planarian, they regenerate and then they stop, right? A planarian just doesn't keep growing and regenerating. It eventually has that off switch. So you have to figure out both switches, but not just the switches on their own, but how to turn them on and off. So it's like the location specificity to it too, where it's like, I want to know the on and off switch, but I also want to be able to control it in space and time, which is an extra layer of variables, Jay, right? That is like, well, you got to figure that out too. But it's all those factors that are going to be needed. Now, we do have that video on the stem cells, and then we have the video on the animal from which they took these genes, which, again, I'm like, when you see this animal, you're going to be like, really? That little creature? Yeah, it's not. It's metaphorically see-through, It's not physically. So shining the laser on our skin bounces right off, so we can't activate genes in a particular region. All right, shall we? The stem cells. This is what had been unable to be made. This is what was a big deal from getting from embryos, where you're able to create, going from that stage, right? From the embryonic stem cells, you could make anything. That's a lot of legal issues and political issues within the country, and I think worldwide it's different places where it is. But obtuse baboon, that is a beautifully said. It's like the ditto of medicine. Yeah. And so here, by switching out those two genes in the mouse skin cells, they're able to push it into this direction. Yeah, we can. We can make from stem cells. You can make blood, skin cells, bone cells, nerve cells, liver cells. Jay, Obtuse Baboon has a really, really good comment there. It's like the Pokemon Ditto. Anything in your body, you can make. Yeah, and Liren, hopefully there will be much better treatments. And with this, you'd be able to replace the bone marrow pretty readily. These tend not to be used, stem cells, to any kinds of research, and there's always fighting on whether or not they should be funded by government money. So the technique that is being used by these researchers in the paper bypasses the need to worry about this, because then there's not going to be anything negative associated with funding of it, because you're not using anything from an embryo. You're taking, and it also, for medical treatments, it would require the best possible match would be your own embryonic stem cells. And a lot of people are just not going to have that. So in order to avoid that being an issue, you just go with a new technique. So this, Jay, is what won the Nobel Prize in 2012, is that they were able to induce this pluripotency. It's good, but it's still not great. And so the science with that gene switching, that's like the next level on top of that. Sorry, now let's pop into, because I promised y'all we should see what organism they took the genes from. So remember we mentioned, and I have this written down because my pronunciation is terrible on this, but it's the cyanoflagellates. They are the closest living relatives of all animals. And again, like we said, their genomes contain versions of the genes SOX and POU, which we know are important for pluripotency. So, Jay, what is the animal that they took these genes from?
A flagellate. Thanks to...
So it is a single-celled animal. And again, like we said, it is the ancestor of everything that is around today, which again, I'm like, that's pretty metal. But Jay, this is the animal they took the genes from to make these stem cells. These little circles here, these are multiple of these cell types. These are the cyanoflagellates. These are the single-cell organisms from the researchers removed 2 genes and they popped it into a mouse cell to replace the mouse version of it. Oh, and how they harvest them. Ah, I see. So these had been just like seen in like pond water, and just checking it out in the water sources. And then when you see them, they went through the process of sequencing them to figure out exactly what these things are. And then they were just sequencing a bunch of other animals, and they found that this is the most ancient, that's still alive today, common ancestor of all animals. So that evolutionary branch of them staying a single cell is what we were looking at there. Multicellularity, single cell, SOX genes. SOX genes are important. We found SOX genes in other animals. Since they are single cell, they don't form stem cells. And then we tested them. There must have been something else to this story, Jay, that we're not getting. That, Jay, is what they grabbed and brought into the lab and took out themselves. Jay, they already had sequenced it. That's the cool part. This was already sequenced. And they looked at what is called the BLAST sequence. So what you can do is you put in a sequence of interest in a database called BLAST. And you see, all right, what else has this gene body? And there could be some differences in nucleotides. So like some of the base pairs might be different. So you'll get variations, maybe a little bit in the structure of the protein, maybe a little bit in the length of it, but pretty much it's the same thing. And then they're like, all right, wait a minute. This animal with a single cell has this gene, and we use this gene to make stem cells. This little animal can't be using it to make stem cells because it's one cell. Now, where I'm lost, Jay, in terms of like what the justification was, like, again, that's why I say they must have known something else, is then it's kind of a big leap to then pop out that copy and pop it into another cell type. Yeah, this is maybe getting a little bit at your question, Jay. Which discovery, Jay? The evolution relatedness. So the actual organism was described in the 1800s, and then there was a researcher that saw similarity between this microorganism and a sponge, and then it kind of gave way to when modern imaging took place. They saw more similarities with like scanning electron microscopy, and then it really was sequencing. So like in the last 10 years, 15 years, that you're able to sequence organisms, probably I guess more 10 years, in an affordable manner. And because you can afford the sequencing of the animals, then you can actually introduce them and like into a phylogenetic tree, so like a tree of life, and then do comparisons on what's more closely related to the next. And so that's how they came up with the split point of us, like all animals, and these single cell organisms, like that split point, making this as the newest organism. Yeah, so Jay, if you take this story through technological evolution and pair it with the current story, right, which is kind of at a standstill, like it's really cool, but you can't yet grow the organs in a dish, that'll continue to evolve in terms of story when they end up having the technology to be able to do that, when the discovery is made of what factors you need in order for the dish organ to develop. They hadn't yet, they'd found, so this was kind of the precursor to like, oh, we found some genes and we're trying to figure out what they do in this animal. It's like single cell. And that at that point, like the genes for stem cells, like that wasn't what the researchers were looking for, because that was not their expertise. And then you had the stem cell researchers come and be like, hey, wait a minute, something crazy is going on here. So that is the organism that they took genes from to make our stem cells, or to make these stem cells and actually grow an entire mouse. Yeah, here again, they've got the phylogenetic tree. They have the breakdown of like where exactly these animals are in terms of relatedness. Copies of the SOX gene. So the size of the letter indicates when it's like the most common amino acid. And then if there's like stuff like, we can't zoom any further. If there's stuff like MRCKQ, then that's a variable set of amino acids, but then you'd always see like the R amino acid in position 2. And they mark where the DNA gets bound, where the protein interacts with a different protein, and those are all maintained as well. They even have like structural stuff in this paper of how the protein would interact with DNA, which is really cool to see. So there's some biochemistry in here in this study, which was really cool to see. There was a lot of sequencing data. There was a fair bit of genetic tricks. Here was that workflow that we looked at, and then making the actual mouse pups at the end, which I think was like the convincing bit of data. Here they did this was like a nice bit of extra validation. So right, if we say that these genes are supposed to interact with DNA, you have to prove it, right? And so here they're kind of making sure that the system works not just on the organismal level of like cloning that mouse, but also they make sure that this choanoflagellate SOX actually binds with human with mouse DNA, and with mouse DNA location sites to make these stem cells. And so these are interaction maps that you can see. And so it's trying to map like, does this protein interact with this one? And you can see based on this is called a ladder. So the dark bands are where there's certain amounts of protein, or you can also do other kinds of plots for DNA and RNA. Protein here and just those banding patterns demonstrate interactions. There's bands for unbound and there's bands for bound as well, and you can tell the difference between the two. So just further evidence for something like that happening. And then of course this is just further breaking down the phylogenetic tree with the other genes. They looked at both SOX factors and the POU, again demonstrating its genetic similarities, just breaking it down piece by piece. And this is kind of the look at how it works with like over evolutionary time. This is the hard part. If it didn't interact, yes, exactly. So if it didn't interact, Jay, there would be two bands at the bottom of the gel for the two proteins being by themselves, or like DNA here and protein. There's still those bands present. There's also the higher up bands. So the lower bands indicate smaller things. The higher bands indicate bigger things. They can't move through the gel as quickly because they're larger. And so when two things are bound together, makes them larger. So you have that double bound band. And then you go lower, and then you have the single ones. But yeah, so like the big takeaway, y'all, I think this final figure is a really nice one, is that before metazoans, so your modern animals, you had a SOX gene and the POU gene, and they're doing two different things. We have no idea what they're doing, but they're doing two different things. And then there's some kind of multicellular creature that starts forming, and there's some kind of interaction with SOX genes and POU genes. Something. And then we have now invertebrates where we know exactly where the interaction is. So there is this, we actually don't know what these two genes that we know are involved in stem cells are doing in this single-celled animal. We have no idea what they're doing. So that's a question mark. We don't know then what that transition animal looked like, where all of a sudden they did start acting together and maybe binding DNA and working together to activate certain genetic elements. And then we have the picture now that makes our stem cells today. And yeah, it's just, I don't know, I think it's a really, really cool paper. Surprised that they haven't really fetched a lot more like of the public media and popular sciences, but again, I thought it was really, really cool. And the rest of it is like a lot of method studies, how they did the sequencing, some of the modeling mechanisms, the cell cultures, how they got the mutant mouse lines, how they did those, immunohistochemistry, all the experiments that they did. They have a pretty good methods section as well. So I'm really looking forward to where this study takes us in terms of stem cells and the development of new treatments based around this. Again, we were talking, it's a couple of steps still to go, but it's pretty far along, which is really, really special.